All right, go ahead and start with the reproductive system for this week. So some objectives that we're going to make sure we cover. By the end of this lecture, you should be able to describe the parts and general functions of the male and female reproductive systems, as well as explain the way in which hormones control the activities of these reproductive organs involved and the development of secondary sexual characteristics. You'll also be able to describe the similarities between the two different systems and explain diploid and haploid cycles of both systems as well. So starting off with the male reproductive system, we have production of sperm occurring in the testes, and we'll talk about some of the anatomical structures more so in lab, but the things that are pertinent to lecture we'll make sure we cover during this section. So when the sperm is produced in the testes, it then travels in the epididymis through this duct system and then through glands. Well, it's through the duct system, and the glands contribute to the production of semen throughout these glands until ejaculation occurs. And so the testes are involved in the production of sperm, while the epididymis, duct system, and various glands are involved in the transportation of that sperm. So the tightly coiled seminiferous tubules form the bulk of each testy. They are composed of developing sperm cells surrounding a lumen in the hollow center of the tubule, where the formed sperm are released into the duct system of the testes. We'll see that on the next slide. Inside the seminiferous tubules are 6 different types of cells, and this includes supporting cells called Sertoli cells or sustentacular cells, as well as 5 other types of developing sperm cells that are called germ cells. Surrounding all stages of the developing sperm cells are the Sertoli cells, which are a type of supporting cell. Sertoli cells secrete signaling molecules that promote sperm production and can control whether germ cells live or die. These cells also create the blood-testis barrier, which keeps bloodborne substances from reaching the germ cells, and at the same time keeps surface antigens on developing germ cells from escaping into the bloodstream and prompting an autoimmune response. The germ cells are stem cells that differentiate into sperm, so let's take a closer look at that. So what we're seeing here is the inside of a seminiferous tubule. This is a cross-section, and the spermatogonium are going to be on the outside of this seminiferous tubule, and they grow or divide via meiosis toward the inside of the tubule in this lumen. So the least mature germ cells are on the outside. They line this basement membrane, and spermatogonia are the stem cells of the testes, which means that they're able to differentiate into a variety of different cell types throughout adulthood. Spermatogonia divide to produce primary spermatocytes, and then secondary spermatocytes, and then spermatid, which finally produce functional sperm. The process that begins with spermatogonia and concludes with sperm is called spermatogenesis. All right? And so what we're seeing here is a spermatogonium, which is diploid in nature, undergoing mitosis into the primary spermatocytes, and then meiosis now creating haploid gametes, and these are secondary spermatocytes that can then form the spermatid. So we're starting with 1 and ending with 4 spermatid. That's how you know we're in meiosis. We're splitting up the genome into 2. And so then we have a bunch of haploid or gamete cells ready for fertilization. Once that sperm is made in the seminiferous tubules, the immotile sperm are moved to the epididymis, or the epididymis. And it's a coiled tube that's attached to the testes, where newly formed sperm continue to mature. As they're moved along this length of this epididymis, the sperm further mature and acquire the ability to move under their own power with a flagellum. Once inside the female reproductive tract, they can use that ability to move independently toward the unfertilized egg. And the more mature sperm are then stored in the tail of this epididymis until ejaculation occurs. Which, when that does occur, the sperm exit the tail of the epididymis and are pushed by smooth muscle contractions through the ductus deferens, also called the vas deferens in older literature. And because these are physically accessible within the scrotum, surgical sterilization to interrupt sperm delivery can be performed by cutting and sealing a small section of the ductus deferens. And this is what's known as a vasectomy, and it's an effective form of male birth control. Although it is possible to reverse a vasectomy, clinicians typically consider this procedure permanent and advise not to undergo it unless you for sure do not want children in the future. Sperm make up only 5% of the final volume of semen, which is the thick milky fluid that males ejaculate. The bulk of semen is produced by 3 critical accessory glands of the male reproductive system: the seminal vesicles, the prostate, and the bulbourethral glands. As sperm passes through the ampulla of the ductus deferens at ejaculation, they mix with fluid from the associated seminal vesicle. The paired seminal vesicles are glands that contribute about 60% of semen, and the fluid contains large amounts of fructose, a nice sugar, which is used by the sperm mitochondria to generate ATP, and this allows movement through the female reproductive tract. The centrally located prostate gland sits anterior to the rectum at the base of the bladder, surrounding the prostatic urethra. We'll cover that more in lab. About the size of a walnut, this prostate is formed of both muscular and glandular tissue, and it excretes an alkaline milky fluid to the passing seminal fluid, now called semen at this point, which is critical to first coagulate and then decoagulate the semen following ejaculation. The temporary thickening of semen helps retain it within the female reproductive tract, which provides time for sperm to utilize the fructose provided by the seminal vesicle secretions. When the semen regains its fluid state, sperm can then pass farther into the female reproductive tract. The final addition to semen is made by the 2 bulbourethral glands, also known as the Cowper's glands, and they release a thick, salty fluid that lubricates the end of the urethra and the vagina and helps to clean the urine residues from the penile urethra. The fluid from these accessory glands is released after the male becomes sexually aroused and shortly before the release of semen. It is therefore sometimes called pre-ejaculate. It is important to note, in addition to lubricating proteins, it's possible for bulbourethral fluid to pick up sperm already present in the urethra, and therefore it may be able to cause pregnancy still. So keep that in mind. Now, testosterone. This is classified as an androgen, which is a steroid hormone, and this is produced by the interstitial or Leydig cells in the testes. It promotes male reproductive system growth in the penis and testes, and the continued presence of testosterone is necessary to keep male reproductive system working properly, and these interstitial cells produce about 6 to 7 milligrams of testosterone per day. Maintaining these normal concentrations of testosterone promotes spermatogenesis in the testes, whereas low levels of testosterone can actually lead to infertility. In addition to intratesticular secretion, testosterone is also released into the systemic circulation and plays an important role in muscle development, bone growth, the development of secondary sex characteristics like pubic hair, vocal cord growth, behavior changes, and maintaining libido, sex drive in both males and females. The regulation of testosterone is carried out by a couple different hormones. So the regulation of interstitial cell production of testosterone begins outside of the testes. The hypothalamus and the pituitary gland, the anterior pituitary gland in particular, they integrate external and internal signals to control testosterone synthesis and secretion. The regulation begins in the hypothalamus with GnRH, or gonadotropin-releasing hormone. The pulsatile release of this hormone from the hypothalamus stimulates the endocrine release of hormones from the pituitary gland, and that would be follicular stimulating hormone, or FSH, or the luteinizing hormone, LH. So both of those are released in response to gonadotropin-releasing hormone. These two hormones are critical for reproductive function in both men and women. In men, follicular stimulating hormone binds predominantly to cells within the seminiferous tubules to promote spermatogenesis. In men, the LH hormone binds to receptors in Leydig cells in the testes and upregulates the production of testosterone. So that's these feedback loops that you're seeing here. A negative feedback loop predominantly controls the synthesis and secretion of both FSH and LH. Low blood concentrations of testosterone stimulate the hypothalamus to release gonadotropin-releasing hormone, which will stimulate FSH and LH, which will help go on and produce more testosterone for the system. GnRH will be stimulating directly this anterior pituitary to release those hormones once again. When concentrations of testosterone in the blood reach a little bit too high or a critical threshold, testosterone itself will go on to bind to androgen receptors on both the hypothalamus and the anterior pituitary, inhibiting the synthesis and secretion of these hormones directly. When the blood concentrations of testosterone once again decline, testosterone no longer interacts with these receptors, and so then they get secreted again, producing more testosterone. The same process occurs with FSH and inhibin to control spermatogenesis. So all of this is regulated. So testosterone is regulated via negative feedback loops. Andropause, might not have heard of this. It's basically the male menopause. It's the decline of interstitial cell activity around age 40 to 50. So if those cells are not working as well, that means that they're not producing as much testosterone. There aren't necessarily clear indications of this, such as menopause with decreased menstruation, which we'll get to shortly. Signs of it can just be fatigue, reduced muscle mass, reported depression and anxiety, which are all related to different hormone loss from testosterone not circulating the bloodstream as well. And then you can do a hormone replacement therapy, such as taking exogenous testosterone, but there's a risk that testosterone is too much in too short of a time, which potentially can lead to prostate cancer, as well as other issues with hormone regulations in the body. So it's possible, and you'll hear people taking testosterone exogenously when they're older in age. There is a risk of developing cancer from that, though.